Our first speaker for this afternoon is Eugene Duff from the University of Oxford, from the analysis group in the fMRI brain lab. Eugene will talk about progress and challenges in large-scale and translational neuroimaging projects.
Thank you very much, and thanks everyone for being here. So yes, I'm going to basically give an overview of the work of the analysis group at FIMRIB over the past 5 or so years, where we've really moved into working with large-scale imaging projects. And you've already heard bits and pieces of a number of these, and from a number of the talks earlier today, from Ben and from Fred, amongst others. So brain imaging really has begun to embrace these large-scale studies and what you might call big data. And our group was a core member of the Human Connectome project, which is probably one of the first large-scale studies of trying to get brain imaging at a much larger scale than had been previously done, in a much more highly systematic approach. We've moved on now and work with on the Developing Human Connectome project, which you heard a little bit about from Ben, and the Biobank Imaging Extension, where we've been helping design and manage the analysis component of. Also, we'll talk a little bit about some work that I've done on combining the traditional smaller fMRI studies to try to enhance the sort of inferences you can make from the traditional studies, in this case to generate pharmacological biomarkers for drug discovery. I think I'm just going to try to focus on a few themes about how this type of work has pushed and continues to push the development of our analysis pipelines, and particularly while it's by no means complete, it really starts to encourage multimodal analysis methods, which I think crucially involve large sample sizes to permit you to be able to make reliable connections between different modalities. And things like quality assurance, which previously in brain imaging has often been a very ad hoc and basically done by hand or by eye. This has really pushed us to better automate quality assurance and to integrate approaches, machine learning approaches which you've heard about before, and well-regularized and sparse methods that can deal with the very large samples. So just to give you a review of the Human Connectome project, which is finishing up now. This was a large project, I think initiated about 6 years ago, I guess, by the NIH. And it was actually initiated by the NIH, and the winning group included a group which consisted of Oxford as well as Washington University, where the head of the whole project was David Van Essen, who's famous for building this map of the connectivity of regions of the visual cortex, as well as a Minnesota group who developed the acquisition protocols, and FIMRIB who were closely involved in developing or providing algorithms for the analysis pipelines. The Human Connectome project involved very in-depth acquisition of brain imaging data from about 1,200 subjects. And these are very long scanning sessions involved at least 4 hours of scanning. So this is a scale that's much greater than had been done in most studies in the past. There was a whole host of different acquisitions were acquired and optimized, including diffusion, resting, and a variety of task fMRI, as well as a recording of a lot of behavioral measures and genetics. It was also intended as a very much an open project, so the data became available quite soon after the initiation of the project, and continues to be as new data is acquired, became available. And this included both raw data and quite heavily processed data, including high-level network models that had been generated by our own tools. And the pipelines as well were also made available. So I think it's quite a standout project that has inspired a number of other projects, including a number which are being driven by the NIH subsequently. I won't go into much detail about the acquisitions, but they utilized developments in particularly functional and diffusion imaging, these multiband acquisitions that really improved spatial and temporal resolution by a degree which really hadn't occurred in the previous 10 or 15 years. And so this enabled very high quality images, the ability to reliably project the data onto the cortical surface rather than simply focusing on largely volumetric analysis, and an acquisition rate that made a lot of typical physiological artifacts much easier to control. The analysis pipelines used a variety of tools, including FSL, but also FreeSurfer, which is a toolkit for projecting data onto the surface, which has been around a long time, as well as other tools and visualization software developed specifically for this project. And certainly I think largely because of the increased quality of the data, pretty much every stage of the processing was revisited and developed in very carefully over traditional processing approaches. And this really, a lot of this is still sort of feeding into FSL and the tools that we provide. An example of this is a tool that we developed right at the start of the project called Fix, which is an automated artifact or cleanup tool. This for fMRI. This is a tool that uses ICA to decompose the data into separate components, and ICA tends to do a good job of separating different neural components and different physiological components from each other. But the Fix tool utilizes a training procedure so that after training on a relatively smaller subset of subjects, it can be automatically used to identify and remove artifactual components. And this is a tool that we now have moved into FSL and can be used and trained up for any new data sets. And you can see that the effects of Fix on this fMRI data here. The first column is just the raw data, and we've chosen quite a large period of heavy motion. Our typical standard pre-processing cleans up a lot of the motion, but you can still see that there's image-based artifacts that are introduced by the head motion. These are things like spin history effects, which are not purely fixed by the simple rigid body alignment that simplistic motion correction will do. So ICA has turned out to provide a real boost to the quality of the data and removing key artifacts. And it's particularly useful in these large-scale projects because of the huge volume of data that you just can't. Traditionally, people might have gone through an ICA decomposition and identified these components, but we really needed this to be an automated procedure so that it can be applied to much bigger data sets like this one. So there's been a huge amount of work, studies using the Human Connectome Project data, and I think it's been a real success in terms of providing a large, high-quality, open-source data set where people can compare methods. This is an example of, or this is a representation of perhaps the headline outcome of the project, which is, I think, still under review, but it's a multimodal parcellation driven by David Van Essen that uses a whole range of different modalities to parcellate the brain into functional parcels. So both structural and functional components are used, including resting state, the spontaneous activity you see in the brain, as well as the task-based activations across the whole suite of tasks that are presented to the subjects in the study, as well as other derived maps, such as this myelin map that we generate from the structural data. This parcellation, which should come out soon, is still a hand-driven parcellation where each parcel is carefully refined based on a careful assessment of a whole range of different modalities. And I think there's a good chance that this may be there's really been a lack in brain imaging of standardized parcellations that significantly go beyond traditional ones such as brokers. So this should provide a new standard parcellation for which researchers can characterize their studies. Another example of some work that's been done in our lab on the Human Connectome project is a study of looking at how we can explore and understand individual differences in brain activity and brain activation. The amount of data required in the Human Connectome project means that we can get a lot of understanding of the individual variability in, for example, the responses to particular types of language tasks or other types of functional tasks. The key challenge until now is that usually the number of data sets are so small that it's very difficult to be able to reliably understand and predict these type of individual differences, and I guess separate individual differences that might be due to physiological factors or to personality factors and these other different levels of explanation. So in this work, Saad Jababdi and his students have explored how he can use how the spontaneous fluctuations you see at a resting state in the brain can be used to make predictions about the individual responses to particular cognitive tasks. So he wants to see how well you can predict the brain activity responses to particular types of tasks simply by looking at a period of resting state in that individual. So effectively what he's doing is some sort of alignment of voxels across subjects that is more driven, that is in part driven by the functional connectivity as well as the structure of the brain. So he wants to learn and make predictions about the activity in particular voxels using not just their location, which you might do in a traditional approach to fMRI analysis, but also look, does the connectivity of this voxel suggest that it's likely to activate in a particular task for this subject. So he uses it as a machine learning approach where he defines over 100 features that characterize the activity in individuals and uses those features to predict the overall pattern of activation so the strength of activation in individual voxels. It's a relatively simple machine learning process. Like at its basis, it's a general linear model, but it turns out to be remarkably successful at making predictions about the brain activity for a particular task based on this generic information that's not associated with a particular task. So here is a task associated with judgment of social states, and these the top row here is for 3 subjects is the true activation generated by this task, and below is the pattern of predicted activation given the resting state fluctuations seen and some other structural features. And it turns out there's very good alignment in general across, or very good prediction based on simply the spontaneous fluctuations in brain areas about their expected role in particular tasks. He's recently, or is at the moment, extending this to disease. So it turns out that this procedure not only works with the healthy brains of the Human Connectome project, but could be applied to a number of subjects who have various types of conditions, including tumours and epileptic tumours. And it turned out that this his algorithm could predict even the movement of, or the relocation or reorganization of brain function after these disease states. And this is currently being built into a software tool where basically you can provide the tool with a resting state scan from an individual, and it can make predictions, and you also provide the structure information, and it can make predictions about the nature of expected activations for particular tasks. This is going to could potentially have a role of applications in experimental neuroimaging. I won't go into this in detail, but other work on the Human Connected project from our group has looked at much larger exploratory analyses which try to identify large-scale patterns in the demographics and lifestyle factors measured in individuals and how that relates to particular features of their brain connectivity. Now you've seen this movie already, so so you know what's happening here. So this is the developmental human connectome project which we're collaborating with Imperial and King's on. This scan was acquired prior to the study, but gives an example of the challenge of trying to translate the human connectome project to babies in utero as well as neonates. And so in the last few years they've been gradually developing analysis pipelines to try to explore similar types of features of babies in utero and babies and neonates. And again, this project is a similar scale, about 1,200 babies in the end, once we've acquired them all. I think we're up to about 200 at the moment. And again, the pipelines that are involved pretty much, again, going over, if you started from the HCP pipelines, pretty much every step of this analysis had to be rethought out to some extent. Obviously, we now have a dynamic period where the babies are of a variable age, so our templates have to be variable across time. It's a lot more head motion and a lot relatively lower spatial resolution. Nevertheless, we're managing to get data onto the surface, and these are very early results, but we're able to find in neonates the existence of these resting state networks and starting to track how they develop over the very early infancy period. Again, you heard briefly about the biobank, which is the other and really the largest of the large-scale studies that we're working on, where the imaging extension of the biobank is bringing back 100,000 subjects for imaging, and we're managing the analysis component of the brain aspect of this. And this is clearly a hugely rich data set where we have a huge amount of longitudinal clinical measures for the subjects. There's 3 dedicated centres here scanning over 50 subjects a day, and so the data, it's been going I think about 6 months through some piloting phases, but already we've got more data from this study than we've had for any study we've ever dealt with. It's a much compressed acquisition procedure compared to the Human Connectome project, about 35 minutes of brain scanning, but we still managed to acquire pretty much all the types of images that were required in the Human Connectome project. And now working on pipelines to deal with this increased scale of analysis. Again, these pipelines, the data is a lot lower resolution than the Human Connectome project, so our pipelines again had to be quite considerably reorganized, and I hope that our pipelines became a little bit more flexible from these large-scale projects. They're certainly becoming more efficient. These ones are largely based on the standard FSL analysis procedures, so extracting surfaces and brain regions and automated functional and diffusion analyses. We don't have a huge amount of results yet from these data, although there's certainly plenty of scope already to do a lot of interesting analyses. We find really exquisite quality or details when you're able to average across 4,000 subjects, even at the group level, even when the data in the individuals isn't that high quality. And so the types of resting state networks we're able to extract do look really attractive when you have this many subjects. At the moment, we're just starting to look at initial relationships with a range of clinical variables. I guess some of the interesting but not unsurprising results are a lot of the key relationships due to things associated with major physiological or clinical measures such as blood pressure rates, and so we're learning to deal a lot better with this sample size with those sort of artifacts. So that's just a small sample of the types of the number of behavioural measures that we're getting. There's a lot of quite curious measures they're recording, how often people eat broccoli or bananas. And so it'll be interesting to see the functional localization or the localization of those types of features. But when you've got 100,000 subjects, I'm sure quite a lot of pretty subtle things, pretty small effects are going to be identified. We're spending a lot of time working on multimodal modelling of these data sets because we do have so many independent measures we can derive from them. So one of our earliest attempts at this is a multimodal Bayesian ICA, which is able to handle appropriately different modality data which has different dimensionalities and different noise properties. And so I think this is it is a very difficult problem to develop robust associations between the different modalities in a general framework. But here this Flickr algorithm does seem quite successful in pulling out major, very broad features that are commonalities across the different types of measures we have. And it's probably interesting that it does show that in many cases it might reveal how what may appear to be functional variation as actually seems to be driven by structural variations in structure and vice versa. So it really makes you think a lot more about the relationships between and interpretations of effects in individual modalities. Finally, and this speaks a bit more to Fred's talk, is some work that I've done on fMRI biomarker identification. And you heard a bit of motivation from Fred earlier about how there's a need to flag or screen compounds at an early stage before they go into the large-scale clinical trials. And there has been some talk that fMRI may be a useful tool for this, where you can perform a relatively small and cheap study to get further insight and to decide whether to put a drug through to a higher stage clinical trial. But fMRI has always been problematic for this because it doesn't really provide quantitative predictions about new studies. People tend to perform small studies, say on an individual compound, and they'll find some brain regions that seem to be associated with the analgesic effect, for example. But it's very difficult to turn that into a quantitative tool that will make a prediction about a new study. So we developed a bit of a pipeline, somewhat inspired by the three pillars that you heard about earlier, that you could use to utilize existing data sets and learn from those signature effects of particular types of compound, and then determine whether these effects, or whether drug effects and whether specific effects to that type of compound, a compound might be present in a new data set, allowing for variations in the acquisition protocol. And so in effect you build a database of existing studies of a compound, and you gain some sort of confidence that you build classifiers that can identify particular signatures of these types of drug action. And then if that signature appears in a new study, you have some confidence that the test drug may be has potential to be have an action similar to those known drugs. This is very much a proof of concept, but we did this on a number of analgesic studies, and we basically explored how well we could identify the presence of an unseen and untrained upon analgesic in a new study based on a training from earlier studies. And it turned out to be reasonably reliable, at least for analgesics. There was all but one of 8 studies we found positive evidence that the compounds were having an effect on the brain, and that this effect looked like an analgesic. I think that a real point of this work, when in the context of this big data meeting, is that it utilizes the traditional smaller studies of brain imaging, but combines them together in a more systematic manner to enable inferences that you can't make from a single study. You can't really do a single study of 1 or 2 analgesics and confidently say you understand what that class of drug might have, what its effects on the brain might be. And indeed, we certainly found specific effects for individual drugs that weren't carried across the general class. And we've been extending this type of work to other applications using resting state, looking at a biomarker for newborn babies, pain in newborn babies that uses EEG and in the future fMRI, as well as a marker for antidepressant action. So just to summarize, I tried to give you a sense for the types of large-scale studies that our group's been working on, and the types of problems that specifically we're facing or working on at the moment. There's no doubt that brain imaging is moving into the realm of big data, and I think most studies in the future will be trying to use the data from something like the Human Connectome Project to provide more robust hypotheses about and quantitative hypotheses about their own data, rather than performing standalone statistical analyses on a relatively small set of subjects. This involves a lot of different components. A lot of it's very low level, making sure that our pipelines are flexible, scalable, and that sort of ironing out these areas which in the past we would let people work with qualitatively or by hand. I don't think the challenge of integration of modalities has been solved. Our group hasn't really implemented complex biophysical models, which is ultimately probably what you'll need to link, say, diffusion and functional brain imaging, or even EEG and fMRI. We're at this point a bit more empirically focused, trying to at least find overall commonalities across the variation in different modalities. We're beginning to focus a lot more on machine learning methods and ways of using sparse or regularized methods to ensure that we are pulling out interesting features at the sort of scale that is neuroscientifically interesting. QC is another thing we've been working on a great deal recently, and I guess the final point is that there's still a real role for these smaller studies, which I think are under quite a lot of heat at the moment from statisticians for some good reason, but I think these being able to perform a very careful, well-designed smaller study in the context of our understanding of, say, individual variability will give them a lot more power. So to finish, I'll just thank the Film Analysis group. I think we work a fairly tight team, and most of us have worked on many of the projects I've described. And just a bit of a plug for the FSL course this year, which is in sunny Sicily, where we take people through pretty much all those modalities analysis that I've talked about, focusing on the FSL package, but also a few other types of tools as well. So thank you.
Thank you very much, Eugene. Are there questions?
Hi, Barry.
So it's a slightly planted question, but what do you see as the key gaps to actually validate some of your integrated techniques?
Which ones?
So the integrated techniques, where you're bringing the data together from your smaller studies, what do you see as the key gaps in that which would make them ready for somebody like a drug company to use?
Yeah, I think, well, I guess one of the biggest things that we grappled with was the variation across studies, and I think the real way that sort of approach is going to develop is for individual labs to develop to plan out a series of studies a bit more systematically, and so that you have relatively well controlled acquisition parameters, but you can start to be more confident in making, combining these separate studies to make enhanced inferences. And there's some examples out there now that do that. I'm always a bit, the large-scale meta-analyses, which there are a lot of people doing those, do have quite strong limitations, so that, and so it is a bit hard to see how people large-scale databasing approaches are going to be relatively limited when you have very specific questions. Okay.
I think there would be more questions, but let's leave some time. Thank Eugene again.